If you've ever walked into your kitchen and you see on your food, maybe a piece of bread, there's this greenish growth. That growth there, that's a fungus, that's a kind of microbe. What we're doing is we're exploring many of these microbes, these single-celled organisms, and asking if we can cultivate them in the lab for specific industrial purposes. One of our scientists, Jennifer Doudna, recently won a Nobel Prize. Her foundational work has enabled the use of gene editing technologies in any organism that you can think of. We have used that research to make fantastically complicated genetic edits to microbes, and that has allowed us to hit product titers that have been unseen before in the past. We're trying to domesticate microbes. We're trying to make them behave in a reproducible and predictable manner. And by looking to characterize their strengths and weaknesses, we are able to identify innate characteristics that make other microbes better for use in industrial processes. If you look out into the Pacific Ocean, we have a small continent growing which is full of plastics. And with biology, we can find ways of producing these molecules that are sustainable. When I walk into Target maybe in 20 years from now, I hope to one day be able to walk in and pick up 5 or 6 items that were produced sustainably using a biological process. That, I think, will be the ultimate goal, the ultimate win for us, where the science we're building here makes its way into everyday life.
I've always been very interested in the scientific concept of energy. And so I really wanted to see how I could contribute to solving our challenge of powering such a large society that is so energy hungry in a way that is sustainable and safe and not damaging to the planet. Right now, even though we have great technologies to capture renewable energy, such as wind turbines and solar panels, only about 20% of our energy comes from renewable sources. And a big reason for that is renewable energy tends to be intermittent. You only get it when the wind is blowing, or you only get it when the sun is shining. Whereas industry and society as a whole needs energy constantly and reliably, 24/7. So my research is looking at ways to store that renewable energy when we have it, hold on to it efficiently for tens to hundreds of hours so that we can then release it later whenever we need it. So we're researching how you can get stable thermal energy storage in materials up to very high temperatures, maybe a couple thousand degrees Celsius, and hold on to the energy that way. And we think this could be the most economical way to store energy at the scale of a city. So I find this exciting because on the one hand, this delves into, fundamental science of how do you make a material stable at these, very high temperatures while still having these required properties of good electrical conductivity, good thermal conductivity, all these different pieces that you need to put together the puzzle of a large-scale energy storage system. And then on the other hand, if it's successful, this could have a huge impact, right? This could let not only the country, but in theory, the world transition to an all-renewable society.
There's probably more bacteria under our feet than stars in the sky. We can observe all of this stuff, and it's all around us constantly. And so if we understand it, we understand how it affects us and how we can use it. I study genomes, and specifically microbial genomes. We usually think about genomes as encoding all the information that an organism has, but it's actually still very difficult to assemble and analyze these genomes. And so that's where I use bioinformatics and computer science to help analyze these and put the genomes together. A lot of my research is in the terrestrial subsurface, so deep underground. There's no oxygen, and then also I've studied microbes that live in hot springs at boiling temperatures, and so it's really inspiring to think that there's life everywhere and that we can study them and understand them through sequencing. Bioinformatics is basically using computer science and statistics to analyze biological data. Like a lot of the biological data that we gather, there's just so much of it that you need the computer to help you analyze it. And so in my research, I actually use the supercomputers here at NERSC and also the cluster here, that's Lawrencium. I think the more that we learn about microbes, the more we can harness their powers for things that we need, like improving agriculture and understanding how they affect our health and the earth.
So I've always marveled at the ability of plants. This is coming from the tiniest Arabidopsis plant to the tallest sequoia. There is this interface below ground where plants, roots, and soil interact. And there's something amazing that's happening there. Millions of microorganisms are interacting with plant roots. My research in part looks at the interactions between plants and microbes and trying to help identify those microorganisms that are helpful and essential to plants, and trying to combat others that are pests to plants without using chemical pesticides. One of the notable discoveries of plant-microbe interactions is actually trying to tease apart individual cells from a microbial community and seeing exactly how they're doing, what they're doing, and how they interact with their environment. An exciting thing about our group is that single-cell genomics technologies have been applied to identify which genes are actually crucial for this interaction between microbes and plants, trying to see how we can actually make other organisms better at helping the plants in this way.
If we start with natural photosynthesis, plants use carbon dioxide, water, and sunlight to produce glucose, fuels for the plants. Artificial photosynthesis is the idea that we would take that same design strategy and engineer it to be higher efficiency and produce different types of products that we would like to use, for instance, fuels or materials to build and power for the next generation. In order to power these devices, we take semiconductors, which absorb sunlight and generate power, coat them with catalysts, which are specific to certain types of chemistries we want to perform, say for instance turning carbon dioxide into ethylene, for example, combine them in unique ways housed in chassis, and then they either get flooded with water or we pump in CO2 with water vapor, and all you have to do then is shine sunlight on the semiconductor and it powers the rest. We seek to understand how to use light in creative ways to power chemistry, to generate electricity. And so what I really enjoy about this whole process is the creative aspect of it all. So working with my peers to come up with creative solutions to problems that all center around how to use light in innovative ways.